Okay, thank you for joining this biology webinar. We're going to be going over chapter 15, the first two sections. And this is on genetic engineering. And genetic engineering is changing the characteristics of an organism by manipulating its genetic material, generally its DNA. Now, humans have been practicing what we can consider a form of genetic engineering on plants and animals for thousands of years in the form of selective breeding. So with selective breeding, there is naturally variation in any population of organisms, and so humans have bred the individuals with the most ideal characteristics, and this shifts the gene pool to contain more of those alleles. And in this way, humans have made some very drastic changes. So for example, we've taken dogs that were very much like wolves, and just using selective breeding, we've created dogs that can run really fast and dogs that can fight badgers and dogs that do whatever pugs are supposed to do. Just over thousands of years of just choosing which dogs we want to mate for the specific traits. Okay, so that's an example of selective breeding and the amazing changes that can come from it. Another great example is corn. So there's a grass that's called teosinte, and that's what it looks like. And over thousands of years of people breeding it, we've turned that into modern corn. So it's the same species. We just made changes just by breeding the bigger ones to the bigger ones to the bigger ones out of each generation and ending up with giant ears of corn. So we show you this to show you the big changes that we've made in both plants and animals just by selective breeding. And something that helps that is hybridization. So hybridization is crossing two varieties of a plant or animal that has a desired trait with a variety that has a different desired trait and hoping that the cross has both traits. So this is usually done with different varieties of the same species. So it's easy to see in examples like mixed breed dogs. So if you take a husky and a corgi and you breed them together, now you have a hybrid that has the short little stupid legs of a corgi, but the facial features of a husky. So you can very much see the traits from each side that got crossed together. Now, this only works with animals and plants of the same species or a very closely related species. So you can't just take things that are completely different from each other, like a bear and a horse, and cross them. That's not going to work. They've got to be either the same species or really close. But it works because you've taken selective breeding to make very distinct strains that are very different from each other, and then you could cross them together and get a combination of their traits. Another term from this section is inbreeding, and that's the continued breeding of individuals with similar characteristics, even if they are closely related. So this is something that has become a problem in many purebred breeds of dogs and some cats and some horses, is dangerous amounts of inbreeding to keep those traits. So the problem with this is that not only will the desired alleles show up more regularly in individuals, but the negative alleles in the bloodlines will also be more frequent. So inbreeding makes it more likely for an individual to get two recessive genes for a disorder. And this shows up again in several breeds of dogs that are prone to problems that mutts rarely have. For example, Cavalier King Charles Spaniels have spinal cord problems, and Dalmatians are often born deaf, and a lot of other breeds have hip problems. And it's because we've really limited the gene pool, and somewhere in that gene pool early on was an allele for these bad traits, and by continually breeding to the animals that happen to have it, it's more likely again that the offspring will have two recessive alleles for this bad trait and end up not being okay. And this is also why humans can't marry their closest relatives. humans that are inbred are not necessarily having any health or mental problems, but if there is a recessive allele in the family for a disorder, the likelihood of receiving two alleles for that disorder increases if marriage and having children happens within the family. And an example of this is the hemophilia in European royalty around the 1900s, the early 1900s. Um, Even though they were different countries, people wanted their family to marry well. So the princes of one country would marry the princesses of another country, and that just kind of kept going until everybody was related. And then at that point, many of the royalty of various European countries had hemophilia because it's a recessive gene, and it was somewhere in the line. And because they just kept breeding to each other, there was more frequent instances of one person getting two copies of that bad gene and ending up having hemophilia. Now, while selective breeding has done astonishing things over thousands of years, there are now other forms of biotechnology that are allowing changes to be made faster. So, with selective breeding, we're kind of just relying on the diversity that's already in the population. And occasionally, there's mutations that happen that add to the genetic diversity and change up the traits that are available. But scientists can also now induce extra mutations and introduce these mutations to the population. So what can happen is they can take radiation or certain chemicals and use that to affect the DNA and make a lot of mutations. And obviously, many of these mutations cause problems, and they don't breed the offspring that have those. But occasionally, a helpful mutation is created. And because it's in the DNA, then the offspring of that individual will have the mutation as well. So this sounds like something that's really weird, but it's something that scientists actually use on bacteria to create hundreds of varieties of useful bacteria. Now, we discussed in the last chapter that having the wrong amount of chromosomes is a problem in humans and in other animals. There should be two copies of each of the chromosomes. However, in plants, it can actually be really beneficial. In many crop plants, this happened long ago, just from mutation or accident. But more recently, scientists have been making this happen on purpose. So they can use drugs that keep the chromosomes from separating during meiosis. So the plant cells go to divide, and instead of splitting the chromosomes in half evenly, one daughter cell ends up with not enough chromosomes, and the other one ends up with too many chromosomes, and they keep the one that has too many chromosomes. And it just creates cells that have extra sets of chromosomes. And that condition is called polyploidy. And again, really not good in animals and humans, but in plants, it usually works out fine. Polyploid plants actually can be bigger and stronger than the regular versions. And a lot of the plants that we grow as crops are actually polyploid. Um, Here's an example. Strawberries that are diploid, like the one on the right, are pretty small, not very tasty. There's different varieties of them, but most of them are not really something you want to grow. But the ones that we most commonly eat have 8 sets of chromosome. Okay, so that's kind of a lot of polyploidy. They have 8 sets instead of the normal 2. And strawberries are not a result of any chemical tampering. It was just hybrid breeding long ago, and they're the strawberries that we ended up with. But they happen to have 8 chromosomes. And again, many, many crop plants are like this. Strawberries are just one example. So that's all of section 1. So we have a quick review question, just based on the last slide. So which is not true about polyploidy? So here's our options. And again, you can't click on anything, just kind of think along with me and then we'll show you the right answer. So which is not true? 3 of these are true and 1 is not true. So is it common in plants but not animals? Is it something that generally creates weak, useless plants? polyploid plants have extra chromosomes, or polyploidy can happen naturally or artificially. So our answer is this one. That's not true. Remember, polyploid plants are the ones that we usually end up using as crops. They create really big, strong plants. And the other three are true. It is common in plants, but not animals. It is extra chromosomes. That's the definition of it. And it can happen naturally or artificially. All of those things are true, except the one that's circled. All right, so section 2 is about genetically modified organisms. So you may have heard of genetically modified organisms, which are organisms in which the genome has been directly altered. Okay, scientists purposely went in and said, we're going to change this gene. And sometimes changes are just made within the organism's genome, and sometimes they actually take genes from a different species and insert it into the genome of whatever species we're working with. Okay, so how is that done? How do they take a gene from one thing and put it in another? Well, it's pretty complicated. Your book breaks it down into a few simple steps, and we're going to kind of go over those. Obviously, this is just a really simple overview. There's a lot of technological stuff happening. But the basic idea is the scientists take DNA from cells and cut it up into pieces by enzymes called restriction enzymes that just cut DNA. So that's what they do normally in your body, and we harness them to do it to the DNA in the lab. Then they use gel electrophoresis to separate those pieces by size so that you've got all those pieces separated. And then we find the one we're looking for. So they do that by using what's called an mRNA probe. So if you think back a few chapters, mRNA, messenger RNA, is RNA that matches up perfectly with DNA. It's complementary to it. And normally that's used to then go out into the cell and tell the cell what protein to make. But in this case, we just have the chunk of mRNA already, and we're using it to match up to the DNA, and that's going to help us figure out the piece of DNA that's the one we want. So the probe, the mRNA probe, has a certain code of DNA on it, and it finds the chunk of DNA that it's looking for, the one that it matches up with, and that's how we know that's the piece we want. So we've cut up the DNA with the restriction enzymes. We use the mRNA to find the right piece, the one with the code that we specifically are looking for. And then we copy it over and over and over by a method called PCR, which we're going to describe in a few slides in more detail. But just know that's a way to copy the gene. And then they take those chunks of DNA and put them into a living cell with enzymes called DNA ligase. And DNA ligase cuts the DNA of the cell that it's in, and allows the new chunk of DNA to be inserted into the genome of the original one. So the cell now has its own DNA plus a chunk of new DNA, and it treats it exactly like it's part of its own original genome. It doesn't know the difference. Okay, and the term for this is recombinant DNA. So it's a new genome which contains foreign genes, and the name for that whole thing is recombinant DNA. Okay, now we mentioned that the way that we copy those genes and make a whole bunch of copies in order to do this is a method called PCR. Now that stands for Polymerase Chain Reaction. So usually we just call it PCR. And the point of PCR is to make billions of copies of one gene so that you can then insert it into other DNA. So how this is done is you have some DNA. Oh, sorry. So again, that you have copies of that template, and it's used to make more copies, and you get billions of them. We'll see the details. So you have PCR in DNA. So we start out with some DNA. Heat is used to separate the two strands of DNA apart from each other. So now we have this naked one half of DNA that's just waiting for other things to come over and pair with it. And we start with primers. Primers are chunks of DNA that mark the places where the sequencing should stop and start. Okay, so those hook onto the original DNA and kind of tell it we're going to start here, we're going to stop here. And then the DNA chunk that we want gets filled in. So it just uses the base pairing rules to match over with the DNA that's already there. And the nucleotides come in, and DNA polymerase comes in and makes a copy of the gene between the primers. So we learned in the last chapter about DNA polymerase. That's just a enzyme that comes and hooks the DNA together, making a DNA that matches the code that's already there. Okay, so we separated it. We used the primers to tell us where to stop and start, and we made this new chunk of DNA. And now that chunk of DNA exists, and it just acts as a template for further copies. Okay, so because of the base pairing rules, you've got this one side of the template. You can make more DNA that comes in on the other side. And now both of those can be used as copies to make more DNA, and they could be used to make more DNA and more DNA. And it just grows exponentially. So each set doubles every time, and then you quickly get to billions of chunks of DNA. So quick review question about that. What is PCR? PCR is used to what? So our options are A, make many copies of a gene, B, destroy extra copies of a gene, C, prevent negative effects from inbreeding, or D, mimic hybridization. I'll give you 10 seconds to think about it. So hopefully you remembered from the last slide that PCR is a process used to make many copies of a gene. So it doesn't do any of those other things. Those are just completely random stuff I put in. But this is what it does. It takes one gene, makes billions of copies of it. And the reason for that, again, is so then we could take those copies and put them into the genome of something else. Okay, another review question. An organism that has its own DNA plus an extra gene inserted from somewhere else has what? The term for that is either A, inbreeding, B, hybridization, C, recombinant DNA, or D, polyploidy. I'll give you 10 seconds to think about it. And the correct answer is recombinant DNA. So again, that term means it's the original DNA from the organism plus an extra gene that got stuck in there that came from something else. So that brings us to the end of this section, and we're splitting this chapter in half because it's a big one. So you can watch the next video. And if you have any questions about this, you can definitely speak with an instructor or talk to us online.